Hello, my name is Mark Muller. I'm the CEO of TopoGEN Incorporated. I'd like to present today our cell-based screening systems for DNA repair, and these were developed for in situ, that is, working within the context of the cell, pathway reporters that tell you about the proficiency or efficiency of DNA repair. This particular video will focus on non-homologous end joining. And this is a major subpathway in animal cells for DNA repair. It's a non-homology based, basically end resection and then resealing mechanism. And it has a dedicated reporter that I'm going to describe within cells. Now this is just an overview of the entire system that I'm going to present. Essentially, we install a reporter that's GFP negative by design. We introduce a double-stranded break at a specific site. This can be with a homing endonuclease such as SCE1, or in some cases with CRISPR-Cas, and we have other mechanisms as well to introduce breaks specifically. The cell then will try to repair these breaks through the process of non-homologous end joining. And we know this to be the case because there's no homology available in order to perform the alternate pathway of homologous recombination. So what I'm going to tell you in this video is focused largely on NHEJ with just some side comments on HR. In about 3 to 4 to 5 days, we start seeing GFP positive cells. And the important thing about these kits is they will allow you to assess NHEJ using specific cell lines. Here's an example. Before NHEJ, we get very low. Of course, if you looked at the entire population, you might see 1 or 2 green cells, very low percentage. And then after NHEJ, we get these cells coming up, and we call them progenitor cells, meaning that they turn green, and you can see the conversion GFP positive after DNA damage over time. And when you see that they appear following a DNA insult, that means they must be undergoing NHEJ. It's important to note that NHEJ also is a rather error-prone pathway, so you don't get 100% successful outcome. In other words, the percentage of NHEJ is generally lower, never 100%. And typically 5 to 10% is the kind of result we would see. It's very important for the application of these kits that you do a negative control to establish background levels of NHEJ. In other words, what percentage? Typically it might be 0.1 as much as 0.5% green cells before NHEJ. Now, the source of this may be from exposure to ultraviolet light or some dying cells that are in the population, but there will always be some low level of green cells or GFP positive cells that you need to be aware of. This can be established in each experiment, and it's important, again, to do this control. This shows an example of the system we use, in this case, with a TET-on induced DNA damage event. So here you see the GFP gene that's been interrupted with some just a random intervening sequence. What this does is it basically insertionally inactivates the gene, so the cells will be GFP negative in this case. But we flanked the insertional element with 2 specific SCE1 sites. Again, this could be a different endonuclease. It could be mediated by guide RNAs. But the key point is that we introduce these double-strand breaks, and we know that the cells contain this sequence by virtue of a selectable marker and also by PCR confirmation. What happens is when you turn on the SCE1 gene with the TET-on promoter by addition of doxycycline, the cells over a period of a few days will undergo NHEJ and you'll get a green readout. Now, we know that the induction with SCE1 is rather robust And this is illustrated in this Western blot showing that within 2 hours after addition of DOCs, we see the SCE1 coming online. So the rate limiting step here is the process of NHEJ. It takes time for the cells to repair. And we're only assaying the wild type GFP that's produced as a result of this. And of course, you're going to see a DOCS dependent production of GFP positive cells as shown here. Now, some of our reporters have a nuclear localization sequence because we're setting up for multiplexing, that is the ability to use more than one fluorochrome in these cells. But here you see the GFP signal is nuclear localized in this case because there's an NLS in the reporter gene. Now the readout is to measure the NHEJ efficiency, or if you will, proficiency of NHEJ. And this can be done by looking at the percentage of cells that are GFP positive or looking at the intensity as measured by flow cytometry, imaging, or imaging cytometry. It is possible to use other means such as a plate reader to measure the amount of GFP being produced in the living cells. This can be TopoGEN cells, an instrument that'll do this, for example. But here you can see without SCE1 there's very little background, and when you add SCE1 the histogram shows in this case over 4% production of GFP positive cells, which is quite convincing that the system is working the way it should. One of the values associated with this technology is the ability to do live imaging, and we can image cells over a period of 4 to 5 days, and we can actually track the appearance of these so-called progenitor cells. Here you're going to see the appearance of progenitor cells. They're just coming on strong here and here. Notice that these cells are in the process of turning green and then dividing. Now this is a process that is ongoing in these cells throughout the cell cycle, which is characteristic of NHEJ. And you can actually track each individual cell into the future. For example, if you pick this cell, or if you were to pick this cell, you could isolate that cell and follow its descendants well into the future in order to ascertain, is there an effect of a signaling molecule, or is there an effect of another pathway protein on the ability to retain the expression level of GFP produced in this process. It's also important to recognize that we have a neutral gene here. So this particular gene, GFP, or these fluoro reporters, are not they're not native, they're not human genes, and they're under no selective pressure. So whatever happens is going to happen without the selectivity process complicating matters. So what are the applications for these kits? Well, any lab that is working on DNA repair mechanisms, looking at gene addition, knockdowns, siRNA, additional novel factors, will find these kits useful. Folks that are working on genes that are important in DNA repair, tangential pathway proteins, testing those for support factors in DNA repair, this kit will find good application there. Looking at repair events during the cell cycle. One thing that distinguishes NHEJ from HR is that NHEJ is a prominent pathway that happens throughout the cell cycle, while HR is S-phase related, typically late in S-phase and G2. Finding druggable targets. You know, there's folks that might be interested in hampering the process of DNA repair in HEJ or HR with small molecule inhibitors or effectors, natural products or other proteins or other factors. The idea here is to make cells sensitized to DNA damage so they don't repair, and this would have value in cancer research, therapeutics, developing new drugs that interfere with the cancer cell's ability to recover from genotoxic events. Many anti-cancer therapeutics are based on DNA damage, so this has real relevance in growth control. Chemical library screens can be performed when coupled either standing alone with chemical libraries and using a microplate approach. This could be converted into a high throughput, or it could be coupled with in silico high throughput screens, and that way pre-select the compounds of interest. So, labs that are working on new drug development would find this kit very useful. Remember, this is happening inside cells. This is in situ. So anytime you get a hit, it has immediate relevance because you don't have to translate that hit from an in vitro situation to an in vivo situation. Right now, we have just a few kits, but we have many others in development. But right now, we can provide human and mouse lines as hosts for both NHEJ and HR. Although this video focused on NHEJ, we also have another video that talks about the HR reporters. We have lineage-specific lines for both NHEJ and HR, and we offer custom services. So we can engineer any cell line as a host. If you have a particular cell line that's relevant to your disease model, you can provide that to us, and we can engineer a reporter for HR or NHEJ or both in a multiplex type assay. Or we can provide you with the DNA and help your lab, your technical staff, develop their own reporters for that. Both of these systems will work. Please contact our technical folks for quoting that service. So obviously this has application for things like disease-specific model lines, iPSCs, lineage-specific lines, and so forth. And this can be done in virtually any species that will support CNV promoter activity, which is many animal cells. If you're interested in ordering any of our kits, please contact us at TopoGEN.com. We'll provide the details. It's important to stress that we have strong product support in our company. All of our products come with consulting, and we make sure that you will apply the products in a most productive fashion for your specific research topic. Any technical questions can be directed to support@TopoGEN.com. For international customers, please see our distributor list on the website and contact your local distributors for more details. Thank you for watching this video.